All right, next topic in Unit 1 is about the scientific method. And the reason why we teach the scientific method is because we want you to have confidence in what we teach you in the biology book. And whatever's put into this book really did follow the scientific method very strictly and is something that we can have confidence in. And we will mimic the scientific method in a classroom, online, or in class to do an experiment so that you can see how the scientific method does work, and it works very well. So our scientific method, this is just in general for students to learn. It's really a lot more complicated than this and a lot more intricate. But just to break it down into its basic pieces, you start with an observation. All scientists are curious and observant. So they see something in nature around them and they're curious about it, and then they question it. After they do that, they probably actually do research. So probably look up and see, has anybody else found out that answer? And if they have, great, and then they can move on. And if they haven't found that answer, then maybe that person would think, maybe I could figure out the answer. And so how would they figure out that answer is by doing an experiment. So before you can create that experiment, you then would have to, with your question, create an explanation or an answer to your question. So if you're observing a plant and you're observing its growth, you might want to wonder, if I added different fertilizer, would it change the effect of the plant growth? So your hypothesis might be this fertilizer is going to improve plant growth or make plants grow faster. And so your prediction is that it would improve it, or maybe that this fertilizer would hurt it. So you have your hypothesis, which is an explanation of fertilizer affects plant growth, and your prediction is that it would increase the plant growth, or your prediction would be it decreases the plant growth. And then you would run an experiment to test that hypothesis. So if I'm trying to find out the effect that fertilizer helps plants grow, then I'm going to design an experiment to test for that specifically. In the end of my experiment, I will draw a conclusion. My conclusion is, oh, this fertilizer does help, this fertilizer doesn't help. Or my hypothesis was correct or not correct. And then you always go back. So in terms of the scientific method, you would do this over and over and over again. So it all goes to also statistics. So it's a class you could take your senior year here at Valhalla. And with statistics, everything has to be statistically significant. So you have to do this in high numbers in order to even mean anything. Doing it once means really nothing in science, except for you did one experiment. But it doesn't get put out into that large body of information, and it doesn't become a theory just because you did it once. So that's something to keep in mind, right? So science, the way science works, if you're a scientist, you would experiment for years, you would publish into a journal, and that journal would then be read by your colleagues and your peers. It'd be scrutinized for how well you did the science and whether or not your results were statistically significant and is acceptable. And in order to become a theory, it's usually not just one experiment, but many people have done many experiments, and then they combine it together. One of the scientists will combine all of these journal ideas together and write some sort of nice, pretty, coherent theory. And then that theory, again, after you read that pretty coherent theory, all the scientists agree to it, and then suddenly we're teaching it to you in a textbook. So it takes years, and it's lots of evidence and lots of experimentation, and observations have not proved it wrong to that point. But scientific theories change all the time because new evidence can be brought forward, and with new evidence, it might change the old evidence, right? Or contradict the old evidence. So we're always changing, we're always growing, we're always open to new things. And I think that you can see that with the coronavirus, because science takes a long time, especially the understanding of viruses, and then developing like vaccines for them. That takes years. And what we've seen is this process of, oh, here's this new piece of information. Everybody gets excited. And then, like the next week, here's another new piece of information, and it contradicted the old piece of information. And some people feel like that means scientists just don't know what they're doing. But no, instead, the scientists were actually sharing with you probably before they should have. They were sharing with you that first experiment instead of waiting until they have done it many times and they know the truth. And so it's really different right now, because we're really seeing the scientific method as it's going along, and it's frustrating because the first time you do anything, it's usually not right in science. It takes a long time before we get it correct. So most scientists run controlled experiments, and that depends upon your scientific field. Biology, controlled experiment is usually something that we can do. Sometimes our knowledge in this book of biology actually just comes from observations over many years and has no experiment. In astronomy, you can't really run an experiment on stars. So again, that will be just observation that leads to that theory. But if we can run a controlled experiment, we will. And a controlled experiment means that you have two groups at least. You can have 3, 4, 5, 6, but you want to have a control group, and then you have your experimental group or groups. So you can like study many things, and you can compare it to the control. The control is the normal. The control is not going to receive the experimental variable. It's what we're going to compare to. That's what makes it controlled experiment. So there's other kinds of experiments out there. We're not learning it in this chapter. So the experimental group is going to be treated exactly the same as the control group, but they get one thing different. They get the independent variable. And down here, this is going to show you a drug test. So this is very much similar, although I will tell you, if it was going to become a drug that we could all take, then you have more than just 500 patients. But we'll just start with this. Like, this is probably the beginning trials. You have 500 patients, you split them up into 250. So your patients should be about the same. And then one group is going to get the drug, the other group is not. Although the other group is going to get a placebo, which we often call a sugar pill. So we're going to pretend like we're giving them the drug if it's a shot or if it's a pill, but it's not, it doesn't have anything in it. We follow those two groups for as long as we have to. And then we find out, did that drug really make a difference compared to the group of people who didn't get the drug? And that, again, would have to be statistics. Is it statistically significant in the difference between getting the drug and not getting the drug? So it has to have significance in math in order for that to be approved. Your control variables are all the things that you keep the same in an experiment. Your independent variable is the thing that you are testing, that is being tested, or is that you're changing in the experiment. So it's the one thing that's different between the control variables is the independent variable. So over here, you can see we were looking at how plants grow, and if you change what you give them besides water. So water would be the normal, right? I mean, I'm assuming you all, if you actually have plants, you know that you water plants. So that's called the normal. That's the control group, right? We're going to give them the normal thing. But then what if we wanted to see, what if I gave them juice instead of water? What if I gave them soda instead of water? What would that do? How would that change how well it grows? Will it grow more? Will it grow less? So we're checking on those things. So our control variables are everything we keep the same. We keep the size of the pot the same. We keep the same amount of soil in each pot. We have the same plant, and we're going to start that plant at the same height. So everything's going to stay the same. We're going to keep it in the same amount of light. We're going to keep it with the same amount of temperature. Those are all of our control variables. The independent variable is the thing other than water. So in this case, it looks like juice or soda. So it might be a little confusing because it feels like we have two independent variables, but really it's just something other than water. And then our dependent variable is what we're going to be measuring in this experiment. What are we looking for? And in this case, we're looking for plant height. So we're going to be measuring how high does the plant grow. That is our dependent variable. It's what we're looking for. It's what we're measuring.